Thank you, everybody, for joining. We'll just give it a couple more minutes just to get everybody into the webinar. All right, why don't we go ahead and get started? I just want to thank everybody for joining us today for our webinar about our new Trinity Freestyle product and how we're going to revolutionize your hybrid capture and targeted sequencing. My name is Rahul Adavia. I'm a senior product manager here at Element Biosciences, and I will be your moderator and presenter today. I'm here with my co-presenter, James Flynn, staff product manager at Twist Bioscience. A few things before we get started. This webinar is being recorded, so if you want to review any of the information presented today, the on-demand version will also be available on our website. We'll also be answering questions at the end of the webinar. Please feel free to submit questions at any time using the Q&A section on the right panel of your interface. We'll try to get to as many as we can, but if we run out of time, we'll follow up with you after. And as soon as the webinar is done, you'll be getting a short 3-question survey. It's your opportunity to let us know what you thought of the content you heard today and any topics you'd like to hear about in the future. So please be sure to fill that out. With that said, let's go ahead and get started. Before we begin, I wanted to give folks who may not be familiar with Element a little bit of background about us. Element was founded in 2017 and has grown rapidly. We've raised over $670 million in funding and proudly hold a portfolio of over 500 patents and patent applications. During COVID, we launched our flagship NGS box, the Aviti, and in late 2024, the world's first and only fully integrated spatial multiomics box, the Aviti 24. Our products are now adopted in over 40 countries around the world. Additionally, Element has been an innovation powerhouse from its inception and has delivered a wide array of impactful product solutions year over year to address customer needs and continues to do so. In 2022, we launched the Aviti with a mission to redefine what's possible on a benchtop sequencer. Since then, we've delivered best-in-class sequencing quality, enabling smaller sample batches while maintaining reagent affordability. The system is high quality, and we've delivered so many innovative solutions with our Trinity and Ultra Q sequencing kits, along with immense flexibility with dual independent flow cells, each with individually addressable lanes. Now, with Aviti and Aviti 24 sequencing, Element has delivered a simple, streamlined workflow. Users can load libraries prepared by their vendor of choice onto the sequencer. These libraries are then circularized before undergoing rolling circle amplification prior to sequencing. This rolling circle amplification produces no founder effect errors, a significant advantage compared to traditional exponential amplification methods. Sequencing on Element instruments is powered by a foundational innovation, avidite-based chemistry, or ABC. An avidite is a dye-labeled polymer decorated with multiple copies of the same nucleotide. The multiple nucleotides on one avidite will bind to multiple active sites in one polony, forming a very stable Velcro-like complex. This allows for dramatically lower reagent concentrations than traditional SBS sequencing, and consequently lower sequencing costs while improving accuracy. So what do these chemistry improvements mean for users? Well, our researchers are seeing Q scores in the mid-40s with our Collaborate chemistry, as you see here in blue, and now in the 50s with our new Ultra Q kits. And so why does this matter? Compared to other benchtop platforms on the market, the Aviti shows clear improvements in read quality, index hopping, optical duplicates, and performance in homopolymer-rich regions of the genome. And these advantages affect nearly every NGS application. The Aviti system was also designed with flexibility in mind. Aviti's dual independent flow cell architecture means users truly get 2 sequencers in one, where each side of the sequencer can be run independently with different start times and run parameters. With the flow cells on the Aviti, each comes with individually addressable lanes, which allows users even more flexibility in their ability to pool two different library pools into the same flow cell. With Aviti 24, we've further expanded the capabilities and sequencing output that we've delivered on Aviti. Aviti 24 essentially takes Aviti and adds additional software and hardware upgrades, along with integrated AI with greater processing power to deliver not just that integrated library prep single cell multiomics capability, but also DNA sequencing with up to 50% higher output using the same kits. So the same kits at the same price, but with 50% more output, dramatically reducing sequencing costs for our users. With Aviti 24, you not only get the Teton cytoprofiling and the in situ multiomics, but also the full array of next generation sequencing capabilities, along with that increased sequencing output. And one of those capabilities that we're delivering on both Aviti and Aviti 24 is what I'm excited to talk to you about today, which is our Trinity technology and how we've expanded Trinity to offer now Trinity Freestyle in conjunction with Twist Bioscience. So before we go into Trinity, I wanted to take a step back and really address what today's challenges are with hybrid capture workflows. As many of you may be familiar, hybrid capture workflows are long and laborious and have several steps and manual, take a lot of hands-on time to complete. Traditionally, you'd start with your DNA library and your target probes of interest before performing a probe hybridization, and then several bead binding and wash steps before doing another post-hyb PCR, QC, and then loading for sequencer. With Trinity, we've really cut down on a lot of those middle steps by either eliminating those steps completely or automating them onto the instrument. With Trinity, now users can take their DNA library of interest, their target probes, perform as fast as a 1-hour hybridization, and load that hyb material directly onto the sequencing cartridge and loading that into the sequencer ready to go. The capture happens on the flow cell directly. And we're really excited to launch this Trinity technology at the end of last year because it really has revolutionized targeted sequencing for our users, enabling less than a 1-day workflow, cutting hands-on time in half, and accelerating time to results, while unlocking 2 times more sample processing capabilities and delivering extremely high sequencing performance. Now we have launched Trinity Freestyle, an expansion to our Trinity portfolio along with Twist Bioscience. What this enables is the ability to take P5, P7 libraries generated with Twist Bioscience library prep, either enzymatic fragmentation 2.0 or FlexPrep ultra high throughput library prep kit with more to come, and hybridize them with your panel of interest and load that directly onto the sequencer with only just a 1-hour hybridization. The sequencing kits will be provided by Element Biosciences, and we've delivered superior performance on this technology with quality scores of greater than 90% over Q30, an on-target percentage of greater than 85%, a fold 80 penalty of less than 1.5, and a mean target coverage of greater than 50x when thinking about exomes. And when we think about exome sequencing, users can expect to run 24 exomes per run on an Aviti or 36 exomes per run on an Aviti 24 with that increased sequencing output. Trinity seamlessly pairs the validated library prep and capture panel from our launch partner Twist with new sequencing kits from Element. So Twist will provide the library prep kits, the adapters and indices panel, along with the hybridization reagents, which includes the Trinity Freestyle binding reagent, which enables this Trinity technology. And from Element, we'll provide the Trinity Freestyle sequencing kit, which contains everything you need to use in order to execute a successful Trinity run. Trinity Freestyle delivers significant advantages against the traditional workflows, as I mentioned before. We've reduced the hands-on time. Because you don't have that post-hybridization PCR, we've really lowered the duplication rate. There's no wash buffer prep or wash or conditioning, no heated wash or manual washes, or any post-capture PCR or QC. And the big thing is there's no blocking oligos needed, which is an additional cost in the traditional workflows. Really, with Trinity Freestyle, we've enabled sample to sequencer in just less than 5 hours. So if you think again about the traditional workflow, you've got your library prep that takes anywhere between 3 to 4 hours. You're setting up your hybridization, and then you do your overnight hybridization, which can take as long as 16 hours. With Trinity Freestyle, that hybridization time is just a single hour, enabling you to start that workflow in the morning and execute a sequencing run that day itself, cutting hands-on time in half, reducing tech time costs, and enabling up to 2 times more sample processing capabilities. Now I'd like to show a little bit of data of Trinity Freestyle comparing to your traditional in-solution workflow. So here we're looking at exome sequencing on Trinity Freestyle, and here we can see comparable or even better results compared to our in-solution workflows. So on the left-hand side, you can see some key metrics of percent Q30, percent on target, and percent target coverage at 30x. And Trinity Freestyle performs slightly better, actually, than the in-solution workflow. But really where we see the best benefit of Trinity Freestyle is actually in the SNP and indel accuracy. When we look at SNP F1 scores and indel F1 scores, Trinity Freestyle performs much better than the traditional in-solution workflow, while delivering approximately the same output of over 1 billion reads on a VD and a much lower duplication rate when we look at the comparison table on the right. Another key advantage of Trinity and Trinity Freestyle is that it delivers highly reproducible and consistent data. By eliminating or automating several of those manual steps post-hybridization, we really cut down on the potential variability that can come from user to user, which results in run-to-run variability. And here we can see across 5 different runs, looking at a 2x150 sequencing run of exomes, that we're able to achieve highly consistent and reproducible and reliable data across the key metrics of mean target coverage, fold 80 penalty, percent duplication, and benchmarking of SNP and indel F1 scores. I'd also like to now give a sneak preview to an interesting application that we'll be showcasing at ASHG this year, which is a PCR-free Trinity Freestyle with repeat expansion calling. So repeat expansions are a well-established cause of more than 50 monogenic disorders, including Huntington's disease, Fragile X syndrome, myotonic dystrophy type 1, Friedreich's ataxia, and several spinocerebellar ataxias, such as ATXN1, ATXN2, and ATXN3. Despite their clinical importance, these loci are often excluded from short-read exome and targeted sequencing panels due to limitations in probe hybridization and the tendency of expanded alleles to have biased representation by PCR amplification, which prevents reliable detection. And in order to address that gap, we've developed a fully PCR-free hybrid capture workflow, which means doing a PCR-free library prep. And because Trinity has no post-hyb PCR, the whole workflow ends up being end-to-end PCR-free. And this allows us to avoid amplification-related dropout and retains accurate representation of repetitive loci. By integrating custom-designed hybridization probes from Twist, targeting repeat expansion loci, this novel method enables detection of pathogenic repeat expansions without requiring whole genome or long-read platforms. So on the plot on the bottom, you can see how we're able to differentiate unaffected and affected alleles very clearly. And on the top right, you can see that the repeat expansion targets added don't compromise core exome performance across the key metrics when we look at this Trinity Freestyle PCR-free workflow with the traditional whole exome workflow. And actually, the fully PCR-free workflow boosts those F1 benchmarking metrics, especially in indel calling as well. But to learn more, please visit our collab at ASHG. We'll be hosting a talk given by Rebecca Barnard from Twist Bioscience, as well as presenting this as a poster at the conference. And with that, I'd like to turn it over to my co-presenter, James Flynn, who will talk about the various applications that Trinity Freestyle can address from agrigenomics all the way to clinical research.
Thanks for Rahul for providing a great overview and background on the Trinity chemistry. So Yeah, that covers the Trinity workflow. I wanted to spend some time today talking about the potential use cases for Trinity and where we're going to take the technology in the future. So, with that, I just wanted to start with some background on Twist Bioscience for those that are not familiar with us. At our heart and our core of the company, we have a DNA synthesis platform, which allows us to create things such as target enrichment panels, as well as oligo pools and other synthetic biology products. And then the NGS side, we also can make synthetic controls based on our platform. So it's based on a silicon wafer platform where we can print up to 1 million oligos in the size of an SBS plate, utilizing our writer technology. It gives us an extremely high uniformity and a low error rate in our DNA synthesis. And we can make oligos up to 500 bases and beyond. And so because we've miniaturized the synthesis of the oligos, it's also a platform which is highly efficient. And so if you compare the creation, for example, of creating one gene and the amount of carbon waste that's produced from that, it's the equivalent on a standard DNA synthesis platform of driving a car 59 miles. Whereas the Twist Bioscience platform and our miniaturized DNA synthesis platform, it's less than a mile, resulting in a 99% reduction in reagent volume and less carbon emitted. So it's a highly scalable platform for us to make different DNA products. And that, as the core, we've built around that a whole NGS suite of products. So really at the core, our platform enables us to play in different markets, such as chemical sustainability. We'll talk a little bit about agbio and how we are enabling food security. We'll certainly focus more on the clinical and human genomics side of things, which allows us to create products for therapeutics and raw materials for diagnostics. And so that I just want to cover generally like what is in our portfolio of products. We make a number of fixed panels, which I'll talk a little bit about today, on how we partner those with Trinity. We make certainly custom panels, and the order of about 4 weeks, we can turn around from the ideation of a custom panel all the way to delivering the finished product. We have a whole suite of library prep solutions, which I'll cover today, and talk a little bit about our future plans of how we plan on implementing Trinity sequencing paired with this. And along with that, we have a number of ancillary products. Certainly, we have a full indices set going all the way up to high throughput, which I'll talk a little bit about today, as well as making fully synthetic controls. Like, for example, we can make a fully synthetic viral genome and have that be as a spike-in control to an assay, as well as for oncology, we make a suite of controls where we can produce a variant at very specific low VAFs, which could be spiked into an assay as a control. So we've been working with Element for some time now, and this is, I think, the third product launch that we've had with Element, where we're really taking our library prep and our panels and enabling different applications through different library prep kits. So we started with an Element native kit, which was based on our EF enzymatic fragmentation 1.0 kit. We've also had the Cloudbreak Freestyle compatibility when that was launched by Element. And then since then, in December 2024, we launched the first iteration of Trinity sequencing, which was based on our EF 1.0 library prep kit. And I'm really excited to announce today that we've expanded this to a new version of Trinity Freestyle, which will, as Rahul mentioned, be compatible with a much broader set of applications. I hope you've been convinced and you've seen now that it really takes on some of the challenges with hybrid capture, where it's a much longer workflow and it kind of prevents or blocks certain applications from being utilized with hybrid capture. So it's kind of a game changer that we now have Trinity. You can do Hybe in as little as 1 hour with really no loss in performance across those. So just to highlight our previous launch, again, we had our EF 1.0 kit. We only had 96 UDIs that were available, and this was based on our standard and fast type kits. And we really paired that initially with just our Exome 2.0, and we've been working out to enable different panels that go along with that. So from that initial launch, we really had a number of asks from our customers is they wanted to utilize not EF 1.0, but our other DNA library prep kits or other library prep kits, such as our RNA-seq kits or methylation. They maybe had novel third-party workflows, maybe single-cell sequencing. And they needed higher throughput, higher capacity, and more content and more applications. So with today, I'm really pleased to announce is that we really have a roadmap where we're enabling new technologies and different library prep kits to be paired with Trinity Freestyle Fast Hyb. So all the boxes you see in gray are now available and validated on Trinity Freestyle Fast Hyb. So our EF 1.0, our EF 2.0 DNA library prep kits, and our FlexPrep kit, which is an innovative high throughput kit, which uses normalization by ligation technology to really scale up the amount of samples you can process. And I'll talk a little bit about how we're utilizing that in the agbio and human population space. We will continue to add additional library prep kits as we validate those. So we're currently working on cfDNA and RNA-seq, and we'll get into methylation in the future. I'm also pleased to announce that we have the FlexPrep normalization by ligation adapters, and we have created a new Trinity UDI set, which is based on our standard UDI 400 set, and we validated all 4 plates. We're currently only have the first plate available, well, we can certainly scale those in the future to have up to 384 indices available for Trinity sequencing. As Rahul mentioned, we also have, it's based on the Fast Hyb kit, and included in that kit will be the Trinity Freestyle binding reagent in there. So with all that being said, what we're really excited to announce is that we won't just have exome sequencing validated out of the workflow, but we're continuing to add new content and available. So I'll talk a little bit about a new panel we've just launched, which is a DNA-based comprehensive genomic profiling for tumor analysis, as well as a population genomics human 600K genotyping panel. I hope people had seen yesterday, we have customers also using this in the agbio space. We have an upcoming release of a genotyping panel for bovine, as well as the suite of all the rest of our fixed panels and custom panel options will be compatible with the Trinity workflow. So I want to go through a couple examples of different applications of the ones that we've currently validated, and then highlight a little bit of the content we have as off-the-shelf panels and how we plan to utilize those to really enable new applications with Trinity sequencing. So with that, our most commonly used exome and our most recent exome is exome 2.0. It's based on HD38, and it includes all of the major genomic databases, as well as non-coding pathogenic and likely pathogenic variants. And we've really tried to go after certain difficult targets, which are included in that panel, including key oncology regions, as well as pharmacogenomic SNPs are included in that. There's a set of sample ID SNPs included in there based on the Pengali publication. And then there's some stats on there. As Rahul mentioned, we have a really high uniformity, a low fold 80 on ours, and a high on-target rate with low duplication rate, which gets even better on the VD chemistry. So this is just showing you a comparison of the metrics for our standard element Trinity kit, which we initially launched, which does not have the Trinity Fast Hyb, and compare that to our 1 hour Fast Hyb. And you can see that the performance is really robust, even with a very fast Hyb. And of course, all of this, with any of our panels, it's always possible to add customization on top. So Twist offers a number of spike-in panels, such as we recently launched a CMD spike backbone spike-in panel, which enables you to do whole exome sequencing, but also do a whole genome CNV calling at different densities of tiling is available. We make mitochondrial spike-in panels, as well as custom spike-in panels. So it's really a flexible platform with our exome 2.0 to do a number of different things for whole exome sequencing. So beyond whole exome sequencing, I mentioned we have something called FlexPrep, which is really targeted to the agrogenomics space. And we had a great presentation yesterday from Hudson Alpha showing how they're using this high throughput library prep to do trait development in the ag bio space. And then it really tackles some things where you want to do high throughput, you want to have high concordance with microarrays, but also have the ability to customize or iterate on your design panels as new markers become available. And it also tackles the high cost of sequencing. So it really makes it a highly efficient platform where you can get the price point down to where you need it to really do these things at scale. And today I want to talk a little bit about the population genomic side of things, where you might be using arrays to look at human genetics at scale. So I mentioned we have a panel that's 600,000 SNP markers across the human genome. It allows you to do a number of things. So I'll talk a little bit about what FlexPrep actually is and what it does. So the UHT means ultra high throughput workflow. And I'm showing you on the top is a typical enzymatic library prep workflow where you dilute and quantify your samples. You go through the whole process of fragmentation, ligation, a spray down and a PCR and another spray down, and then you dilute and QC those libraries. So just like Trinity abbreviates the hybridization workflow, FlexPrep is really abbreviating the whole process of doing library prep at scale. And it's based on a technology we call the NBL, or normalization by ligation. And this really eliminates the need to do upfront quantification and normalization. So you just go right into fragmentation. And in the ligation step, you're actually barcoding the samples across the row of the plate. So you're barcoding 12 samples across the row. Those 12 samples then get pooled together. And then so you've essentially collapsed your 12 samples into just one tube. And so that whole 96 sample plate is collapsed down to an 8 well strip cap tube. And then you don't need to do any additional normalization. So now I'm showing you some data on FlexPrep, where this is not based on Trinity, but I'm showing you just as an example of what you can expect to see when you're preparing these libraries. Is you can do a mass input of 30 to 300 nanograms. And then that's on the x-axis. What I'm showing you on that top bar plot is across all of those inputs, you really get the percentage of read representation is very even with that varying the amount of input. So it really takes the tedious part of the upfront quantification, and it also allows you to process quote unquote dirty samples, which you typically see in agbio, or challenging samples in population genomics. And so across all that mass input, you get just a really clean way to do a high throughput, and by eliminating some of the really tedious manual steps in the process. And how we're utilizing this is to really do population genomics at scale. So in the top left there, I'm showing you an example of concordance with a very common human population genetics array. And you can see with as little as 1 gigabyte of sequencing with imputation, you get really high concordance, greater than 99%. So we've set this workflow up so that we can have the library prep with FlexPrep. You can go right into target enrichment with Trinity. And on the back end, you can use imputation with some of our software partners, such as GenCove. We have other software partners as well who can do this. And on the bottom, I'm showing you the performance you can expect. It works really well. So this is an example of a run where we've plexed 96 samples into a single VD run based on our human genotyping 600K panel. So it's really an efficient solution to kind of scale up your research. So I kind of covered a couple of application areas that we've already validated and are available today. I wanted to spend a few minutes talking about where we can take this technology and different applications that we can really, enable some really neat things with that. So recently, a little bit earlier this year, we just launched a CGP panel, which is really a panel that's focused around just the key attributes that you'd want to see within a tumor sample to understand what's going on in that tumor, and then understand from a research perspective which potential therapies could be informative. So it's a great research tool to look at different genomic alterations as well as genomic signatures. So it can do SNVs, it can do insertions and deletions that are relevant to cancer, certain copy number alterations, and then also certain gene rearrangements as well can be targeted. And so it measures tumor mutational burden and MSI as well. And why we launched this panel is when we looked at the market, the most common CGP panel in the market was launched in 2018 by Illumina. Since then, that panel has not really been updated. Even in this year, they launched a new version of this kit, but the content hasn't really changed. And on the top, I'm showing you a quote from a publication where they said approximately 43% of all precision oncology therapies use some sort of biomarker testing. So and the plot I'm showing you there is the cumulative number of additional FDA approvals where biomarkers were used year after year that accumulates. And so we've had a number of customers come to us and ask if we could make a similar panel, which would also enable them to customize the content and add to it and iterate it over time. So that's exactly the use case. We have a number of customers utilizing this panel. And they've kind of taken that and they've added the specific biomarkers that they wanted to have in their research studies so that they can really, look at novel things in cancer. So this is just showing you again, like what's included in that panel. It has 562 genes, but it has the additional customization option on top of that. And it's paired with our EF 2.0 workflow. You can certainly use it with other library prep kits. We have not yet validated that on Trinity, but that's something some of our customers are working on to utilize this panel plus Trinity plus doing it on either solid tumor or cfDNA. And one other bottleneck that comes up frequently is how do I analyze that data and get to meaningful results? Consider there's not just SNV calling, but you're also looking at genomic signatures. So we've partnered with a number of commercial software providers from QIAGEN, who also is now providing the Genox solution, Franklin. We're paired up with Sequon. Sequon will be talking a little bit about their solution at ASHG and how they're pairing our panel and chemistry with their software solution. Twist is also in the process of building our own open source pipeline. So if you don't want to buy a commercial solution, Twist will offer free of charge an open source pipeline that we've been working on with Fulcrum Genomics. And Fulcrum Genomics will also, and QIAGEN, all of these partners will be at ASHG. If you want to come by our booth, we're happy to chat about that. This is now showing you a little bit of data that we've generated. This is very new. So we have the sequencing metrics here. We haven't taken this all the way through one of our software partners to do the variant calling analysis. That's in the works, and we hope to have that in the coming weeks. But this is just showing you from our 2.4 MB panel, what we did was plexed a 2x12 plex run. So 2 different hybridizations of 12 tumor samples. We plexed that into a single flow cell on the Aviti with an output of 950 million reads at a Q30 of greater than 94%. And we get about a 1500x mean target coverage. And this is based on 50 nanograms of input. So very excited for this. We think it's going to be a great solution to do really rapid tumor analysis where you can, make your library preps in the morning. You can run up to 24 tumor samples on one side of an Aviti and get an output in a really rapid fashion. And then you can continue to do that, day after day, and your throughput would go up as the week goes on. So those are sort of some applications we've covered. I want to talk about one other future one that we're also working on is that Twist makes a comprehensive viral research panel. And what this is a very large panel. It has over 1 million unique probes that are targeted to greater than 3,000 viral genomes of both single-stranded and double-stranded DNA and RNA viruses. So we have a whole protocol and workflow for this library prep, so you can capture both the DNA and RNA viruses into a single assay. It spans both human and non-human viral pathogens. And as I mentioned, we're not just in the human space, we're also in the agbio space. So if you had a particular virus that's not included in this panel, we can always create a custom spike in, or if it's a different variant. We've had recently things like last year, the H5N1 influenza outbreak. Even though this panel wasn't designed to that specific variant, what it did was it still captured some of that information, but we also did create a custom spike in for that particular isolate of virus. So it's a very flexible platform to look at viral research. And this is now from a publication that was looking at different methods utilizing untargeted metagenomic sequencing on either a long read platform or a short read platform, and then compared that to the hybrid capture version of our platform that utilizes both the DNA and RNA viruses. And what you can see for some common viruses is that at a much lower genome copies per mil, we can get a much higher percentage of genome coverage. So essentially the titer that we can see is much better than what you can do with just untargeted metagenomic sequencing. So it's a highly efficient platform. And then what I think is going to be really great with Trinity is with that 1 hour Hive workflow, we can do this really rapidly. So if you're interested in assessing a sample and wanted to understand which viruses are in there in a comprehensive fashion, it's going to be a really great platform. We think this is going to really open up some potential use cases to rapidly identify viruses in a sample. So those are just a few of the potential use cases that we have for viral sequencing. We're going to continue to expand upon the Trinity platform with different panel content, different library preps, as I mentioned. So there's going to be a lot more to come, and we hope as we get into conference season here to be sharing a lot of that as it's available.